We have seen that neurons can express a wide variety of different types of voltage-gated ion channels, and these channels can have widely varying properties. Depending on the complement of channels that are expressed in a neuron, that neuron can have different types of electrical behavior. For example, motor neurons tend to generate single action potentials at a time at relatively low frequencies. Thalamic relay neurons, in contrast, generate rhythmic bursts of activity. Inhibitory interneurons tend to generate high frequencies of action potentials, and modulatory neurons, such as those releasing acetylcholine, serotonin, or norepinephrine, generally have an ongoing spontaneous activity at a relatively low frequency. Again, these types of electrical behavior, spiking behavior, arise due to the different types of voltage-gated channels that are expressed in each type of neuron. This slide illustrates the responses of neurons expressing different types of ion channels and demonstrates that the behavior of the neurons depends on the ion channels that are expressed. For instance, if we look at panel A here, this cell expresses just the standard fast and activating sodium channel and the delayed rectifier potassium channel. Now, when the cell is depolarized by a current pulse, which is not shown here, this generates a steady string of action potentials. The currents from the sodium channels and potassium channels are shown below. Now, if in addition to the sodium and delayed rectifier potassium channels, we add a calcium activated potassium channel, which is shown in panel B. This is the BK type of calcium activated potassium channel. We see that when an action potential is generated, there is an enhanced after hyperpolarization, and this tends to slow down the frequency of spiking. If we go to C, in this case we've added a transient potassium channel, or an IA current, and when the cell is depolarized, we are activating the IA current. This increases the total potassium conductance and slows the rate of depolarization, and this results in a delayed firing of action potentials. If we go to panel D, in this case we're adding an IM current. This is a current we haven't talked about before, but what is happening is that muscarinic receptors lead to an increase in potassium conductance. An additional potassium channel is open, and as you would imagine, when this happens, the current will lead to a reduced depolarization because there's additional potassium conductance in the cell, and this leads to a decreased response to this current pulse and depolarization. If we move on to E, panel E, in this case we've added the other type of calcium activated potassium channel, the SK channel, and once again we see that the after hyperpolarization is enhanced, but in this case the after hyperpolarization increases and lasts longer for each spike, and this leads to a frequency adaptation where each spike is delayed further from the last spike. If we go to panel F, in this case we're adding the T-type of calcium channel, the IT current, and what we see occurring here is that when we depolarize with a current pulse, this triggers a calcium spike. We're opening up the T-type calcium channel. This produces a delayed and prolonged depolarization, and superimposed on this calcium spike are several sodium spikes. So what we've generated here is a burst of spiking when the cell is depolarized. Now, interestingly, if we depolarize the cell, if we maintain the expression of the T-type calcium channel, but we depolarize the cell, what we then do is inactivate the T-type current, and so the channels cannot be open, the calcium channels cannot be open when we depolarize, so we don't see bursting behavior, rather we just see isolated spikes that are generated. To end today's lecture, I'd like to consider how combinations of ion channels lead to oscillations and bursting behavior in neurons. And we're going to specifically consider oscillations that are generated by neurons in the inferior olive. Inferior olive neurons project to Purkinje cells in the cerebellum via climbing fibers, and when these cells are activated, this leads to the initiation of complex spikes in Purkinje cells. Now these olivary cells that project to Purkinje cells fire in synchrony. There are many of them in the inferior olive, and they are all coupled together, so they fire synchronously at about 10 hertz. And when this occurs and the synchronous bursting behavior activates the Purkinje cells in the cerebellum, this is thought to determine the timing and organization of motor sequences that are controlled by neurons in the cerebellum. It's known that the olivary cells that project to the cerebellum express a number of different types of voltage-gated ion channels. These include the fast inactivating sodium channels and the delayed rectifier potassium channels. Together these generate the sodium action potential. But in addition, these cells express low threshold calcium channels. These are the T-type calcium channels. The cells also express calcium activated potassium channels, specifically the low conductance SK channels. These are the apamine sensitive channels. And finally, these olivary cells express the hyperpolarization activated cation channel that generates the IH current. These are the HCN channels. We will close this series of videos with one additional exercise that we'll do in class. We've seen that the olivary cells express a number of different types of voltage-gated ion channels, which are listed up here. And these cells generate a characteristic bursting behavior that is illustrated in the graph down here, where the cells have a slow oscillation that repeats about 10 times a second, and superimposed on this slow oscillation are a series of very fast action potentials, so that this cell will generate bursting behavior, burp, 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 where a burst of spikes occurs 10 times a second. So, given that these cells express these types of channels, I'd like you to think about how this type of behavior is generated by these voltage-gated channels. And then in addition, I'd like you to draw the current flowing through each of these types of channels as a function of time. So superimposed on this voltage trace, I'd like you to draw the currents going through each of the types of channels.